Hello, today I'll present results from our research study regarding the identification of citrullination as a bona fide post-translational modification of TDP-43, as well as discuss the implications that it has in late and AD pathology progression. As we all know, the wealth of study has demonstrated that TDP-43 pathology is one of the hallmark pathologies of Alzheimer's disease. Moreover, the studies within the last couple of years have demonstrated that TDP-43 pathology also contributes to late, the limbic predominant TDP-43 encephalopathy. Moreover, the TDP-43 proteinopathy in late progresses in 3 stages from amygdala to hippocampus and then cortex. Importantly, the TDP-43 proteinopathy contributes in cognitive dysfunction and cognitive decline, both in AD and late, particularly in the brain that coexists with other proteinopathies such as amyloid beta, tau, and alpha-synuclein. In our laboratory, we looked at post-translational modification and particularly citrullination and its role that it has in TDP-43. Citrullination is a protein deimination as a result of enzymatic activity of the peptidyl arginine deiminases or PADs. Citrullination is irreversible and implicates the removal of a positive charge from arginine to a peptidyl citrulline, which is a neutral amino acid. This change of the amino acid and moreover the charge distribution contributes to changes in the protein properties, stabilization of alpha helix, and therefore a denaturation and unfolding of the protein. Both in physiological conditions and in disease, citrullination of proteins have been described. In the case of irrelevant to CNS disorders, citrullination of proteins has been found both in Alzheimer's disease and ALS, as well as induction in protein levels of PADs. PAD4 is one of the enzymes of interest for our research as it's found both in neurons and glia. Our initial data indeed confirmed that the mRNA levels of PAD4 gene were induced in AD brain tissue compared to controls. Moreover, we confirmed the protein expression and found that PAD4 protein levels are induced in Alzheimer's disease brain tissue with the presence of TDP-43 pathology, as well as induced in late 2 and late stage 2 and stage 3 brain tissue, postmortem brain tissue. Interestingly, we also wanted to ask the question whether, based on this data, whether PAD4 was able of citrullinating TDP-43, otherwise a completely unknown post-translational modification of TDP-43. As such, we performed a simple enzymatic reaction between PAD4 and TDP-43 full-length recombinant protein. The reaction was analyzed by mass spectrometry, which identified that 11 out of 20 arginines were citrullinated on the TDP-43 protein. Here we represent the spectra of one of the sites, the citrullinated site, the arginine 83. That prompted us in producing specific antibodies against the citrullinated sites of TDP-43. In fact, we have generated 5 antibodies out of the 11 sites as distributed throughout the regions of the TDP-43 sequence. Our identification and characterization of these antibodies in the recombinant protein system identified these antibodies as specific to its citrullination site. Moreover, I wanted to highlight the presence of the high molecular weight species of the citrullinated TDP-43 identified by these antibodies. Next, we'll ask the question whether or not citrullinated TDP-43 is a bona fide and exists in disease states. As I mentioned, citrullination of TDP-43 is not described in the literature. For that, we return to the postmortem brain tissue and looked at both Alzheimer's disease brain tissue as well as late, where we confirmed the presence of citrullination in Alzheimer's disease with TDP-43 pathology, as well as we demonstrate for the first time the presence of citrulinated TDP-43 early in late progression at stage 1. This is amygdala tissue. The data were further confirmed by biochemical assays, where we demonstrate that citrullination of TDP-43 at different sites exists, and more importantly, is significantly increased in Alzheimer's disease brains with TDP-43 pathology. We also found that this citrullination of TDP-43 with the sites that we investigated were also significantly increased as the late disease progresses through stage 2 and 3. To note, the TDP-43 pathology is confirmed by the positivity for the phospho-TDP-43. We also turn to the transgenic animal models that we have in our laboratory, and this is the TAR transgenic animal model that overexpresses the human wild-type TDP-43 under the Thy1 promoter. We here confirm that PAD4 levels were increased both in heterozygous and homozygous mouse model, and we demonstrate a gene dose-dependent increase in the PAD4 expression levels. Looking at citrullination of TDP-43, we confirmed that this increase in PAD expression also translates an increase in citrullination levels in the homozygous mouse, which also represents the most severe progression of the pathology. This is a quantification of the biochemical data. We also demonstrate the profile of citrullination of TDP-43 in the TAR animal model, both in heterozygous and the homozygous model, confirming the presence of citrulinated TDP-43 in neurons in the homozygous TDP-43 transgenic mouse model. In conclusion, I hope I convinced you that we show that PAD4 levels and RNA levels, protein levels and RNA levels are increased both in patients and TDP-43 transgenic animal models. We show that TDP-43 is a substrate of PAD4 activity and have generated novel specific citrullinated TDP-43 antibodies, where we demonstrate the accumulation of citrullinated TDP-43 in the brain of patients and transgenic mouse models. Finally, the data not shown, our preliminary data demonstrate changes in citrullinated TDP-43 aggregation profile. With that, I want to thank members of my laboratory, our collaborators span within different institutions in the country, as well as my funding sources. Thank you.